Hi, thanks. So today I'm going to present a kind of experiment that we did to match graph theory and time series. So the idea of visibility graphs is that they were defined a while ago in computational geometry and computer science as a graph capturing the visibility between different points. So we can have an analogy with the landscape, or in 1D like that, we can have the visibility between peaks there. So how do we actually build this visibility algorithm? So if you have a time series of 20 points, we can build for each time point these visibility graphs, checking whether one point is visible from another one. So there is actually a small formula to evaluate this, but this is the basic idea on how to construct these graphs. So this is what we obtain eventually if we apply the algorithm to all the graphs. So what are the properties of these graphs? The visibility graphs of a time series remains invariant under several transformations, so stretching or squeezing the time series or adding a trend, and so on. So this is an important property. And of course, so we obtain from a time series, we obtain a graph in which the nodes over a graph are time points, and the links between them are according to this visibility criterion. Then, what happens if we go to a multivariate data sets? With multivariate data sets, we have multiplex networks or multi-slice networks. Now it's a concept which is quite ubiquitous. We have seen already some examples in this conference. And so if we want to compute dependencies across a multivariate time series with the visibility algorithm, we can compare these layers of this multi-layer network using mutual information. So why apply to fMRI? So our idea was that we could test this informative approach, which is furthermore robust to processing and quite easy to implement. And then, out of curiosity, to build a bridge between time series and network and apply to fMRI. Why specifically to fMRI? So a bit more specific motivations. We know that peaks of bold signal encode relevant information on coactivation and correlated activity. So there were a couple of papers in the past years. Yesterday we also heard from Xiao Liu, who talked about these coactivation patterns and presented a recent review. And so basically the idea is that relevant information on the dynamics of fMRI is encoded in these peaks. And since the bold peaks are the ones who are most visible in fMRI, so we thought that we could have built this visibility graph from a bold time series. So this is an example. So this is what we obtained for 3 regions of interest, just an example. And the points are color-coded according to time. So what we did, we applied to a large OpenFMRI data set, the UCLA LA 5C study with healthy controls and 3 mental disease samples. And then what we did is then to compute the visibility graphs for each region of interest, and then we average within the intrinsic connectivity network defined by Thomas, and we did this for all the subjects. So this is what we obtained. The average mutual information across resting state networks, this is what it gives for the 4 groups, control, schizophrenics, bipolar, and ADHD. So we have already some variability across networks. The network in which there is less mutual information, despite being small, is the limbic system, this one. And the limbic system is also the one which separates the 4 groups. So this is it separates the groups between schizophrenic and control across all the deciles. So this is a shift function which is used to compare groups rather than bar graphs or whatever. So what is similar to visibility? We, thanks to NeuroVolt, in which we could compare the visibility maps to other already published results, we saw that as a similar expression across the brain as regional homogeneity, and in the limbic system we also have a higher coefficient of variation of the Bolt signal, and there is also a lower value of the fractional amplitude of low frequency fluctuation. So this could speak more or less to the same phenomenon. Now, if you remember from the previous figure, the visibility graphs are modular, so it means that exhibit the presence of subnetworks. So what happens? When we have modules in a temporal networks, they correspond or they speak to different temporal regimes. And different temporal regimes have been discussed widely in this conference and in the past in the framework of time-resolved functional connectivity. And so we tried to apply this framework now to visibility graphs, comparing visibility graphs in terms of their modular structure. How do we do this? Using a global measure, which is the partition distance, and the local measure, which is the Sorensen index, which was originally defined to compare ecosystems, so to compare the number of species present in both ecosystem C and ecosystem B. And we did the same, basically, using the links into visibility graphs. So this is, for example, what we obtain in two regions of the default mode network, the anterior cingulate cortex and the posterior cingulate cortex, which are split in different clusters. And in this case, we have a global measure, which is the partition distance, and then we have a cluster by cluster similarity measure, which is the Sorensen index. So if we do this for each pair of links, we can compare, we can obtain a measure of time-resolved functional connectivity basically in a single snapshot. This is what we obtain applying this framework to a large data set of scans which were already analyzed in terms of reproducibility of time-varying functional connectivity pattern from the lab of Vince Calhoun. And so this is what we observe with the standard Pearson functional connectivity, and this is what we have from the visibility graph mutual information partition distance. And we see that this measure is, once we standardize, because they have a different range of values and so, and z-scores, and after also Box-Cox transformation, we see that this is more reliable across subjects. And also, if we apply a singular value decomposition, we see that while for Pearson functional connectivity we need a few components to explain all the variance, basically all the variance is explained by the first component with this approach, meaning that the dimension of variability in time is captured in this way. Now, in order to check whether there was some ability of separating clinical feature. In this case, we used another data set from Vince's lab, in which they showed that functional connectivity, dynamic functional connectivity was better able to separate schizophrenic and healthy controls. And what happens? While for the static mutual information visibility graphs, we reproduce the pattern of separability between schizophrenic and healthy controls, we don't reproduce it with the dynamic version, despite the fact that it's very reliable across subjects. So what can this mean? It could be that, let's say, this approach captures a common backbone across subjects, which can be connected to to physiology or movement or so, but that the specifics, phenotypes of this particular mental disease are not captured in this way. So I thank you for your attention. If you want to play with the code, it's here, and thank you.